Alright, okay. So, hello dears! Ayan, and welcome to our first part, no? The first part in our lecture in Virology. Alright, so for this part, medyo ano lang muna tayo, pre-recorded. Okay? Sorry naman. So, ayun, no? So we're down now to the last part na gyud of microbiology, and that is your virology. So we're done with para, which is again, separate na siya with micro, no? And then of course, mycology tapos na rin, and then finally virology. Of course, Bacte was already handled by the queen of Bacte, yes naman si Dean Rodriguez. So, I hope daghan mong na-learn from her because ako po daghan pong na-learn and nag-review po ko na-review ko og ahat like okay. Kaya medyo na TMI sad ko atong dapita pero na-shock sad ko abinahog. Abi na ako, okay pa akong bakte, pero pagbalik ni Dean kay Murag, shucks, asa na to tanan, wala na. But anyway, ayan, so we're down now to virology, no? So as I've mentioned already to you, dears, sa review season, and even sa board exam, no? Virology together with mycology, sila yun ang mga neglected na areas or aspects sa microbiology. Kay ginatagan na sila gamay na time, no? Dili na nakadaghan. Uh, or di sila gina-effortan inana. So I can relate. Charot. Tsaka lang. Ayan. Ayan. So sila yun ang mga ano lang, gina-neglect mga isinasantabi. Ayan, nasa basta neglected yun siya. But anyway, it's a boards pod again, medyo dili kay daghang questions mo gawas from Myco and Viro. Usually mga 1 to 2, kinadaghanan ng 4. Oh, pinadaghanan ng 4. Sa akong boards, ang nigawas 1 ra ka Myco na question and usa pod ka Viro na question. So nakachika na ko sa unsa na Myco na question ang nigawas sa atong lecture sa Myco. Ako pong ichika kung unsa sa Viro. Number 1 si Viro, number 100 si Myco. Inana ako ang exam, o di ba? Sa ako ang board sa micro para. So duha ra gid siya. So inana siya ka neglected o dili kayo ginahatagan og time. But because siguro of this pandemic in the future in your board exam baby or in the future basin mas daghan ng questions na magawas sa Viro or sa Mycopod. So, paghandaan natin yan. Okay? So, again, heads up lang po, dears, atong lecture. Dili gid siya katong inato ka in-depth. Alright? So, more on reviewing lang siya, no? Glossing over and emphasizing the things that are important or those that are mugawas gid sa boards. Alright? Because again, in the review season, usually half day rani, no? Half day, half sa last day sa microbio na part ang imuhang mycoviro. Dito na siya i-cramp tanan. Alright? But for us, at least, ato siyang gihimo na 2 days. Di ba? So, ato siyang ginatagan og time at least 2 days. No? Pero again, it's not in-depth gyud kaayo gyud na kanadyo magdugay ta sa mga topics, kay mag-discuss gyud tawag intense. We're just going to gloss over lang and emphasize the stuff. Alright? So, para di mo mag-expect. Because ako pod mismo, medyo apiki sad ko diring dapita, dears. No? Sa Viro, medyo scary pod akong na. Medyo scary ang Viro actually. It's quite complex to understand. Munang medyo dilipod ko na naka well-versed yun. Pero I'll do my best pa rin to explain and to discuss. Alright? Especially those that mugawas good sa boards and those that are really important. Alright? So again, muna siya ang clinical virology nato na lecture. Alright? Daming chika, no? 3 minutes. Nagamit na ako siya for chika. But anyway, so of course, before we start, unsa na mga references akong igamit, majority are mga microbio na books. Alright? So similar references with the ones I used sa atong mycology na lecture. Majority sa Viro noon akong gikuha is from kani Murray na book. Kani siya na book, dear. It's really good. It's really great, especially for Myco and Viro na part kay Chada iyahang chapters. Iyahang gi unsa ni basta in-depth iyang discussion. So if you want to read more, I suggest Murray. Alright. Tapos I also got from my review notes sa Lemar by Ma'am Vanessa Rivera, yes, top 1 ni siya sa board exam February 2017. Some information from Bailey also and from Mayhon. Ang Bailey manggod kay medyo TMI, dagan kayong mga chika. Ang Mayhon, chadara po siya, it's very short pero concise po, so chadara po siya. So it's a mixture of these references. Gamay lang from online resources kaayo, more on the textbooks. Alright? Okay, so that's for the references. So you can choose any of this. Akong ma-advise jud is Murray. Nice kay ang book na Murray for virology and mycology. I think same yung book na gigamit ni Ma'am Fudge. Review ko sa akong mga handouts sa una sa third year sa virology. Same yung mga gikuhaan og stuff, no? So murag Murray ang ginagamit ni Ma'am Fudge for Viro. So it's really good pod. Okay. Alright, so ang daming chika. So those are for the references. Okay. Now we go now to the lecture proper, we start first with the viruses. So what are viruses, no? Your viruses, very important lang or very unique characteristic to them is that they are not cells. Okay, so please make sure yun nakabalo mo ana or like it's well established na your viruses are not cells, no, rather they're actually called particles, no. Unlike your fungi, parasites, and even bacteria, they are cells. They are called cells or they have cells, good, no. But for viruses, they are not cells, they are called particles. Okay, and first of all, they were considered to be filterable agents because in the past, those that are used, no, filters that are used for bacteria, these viruses can pass through these filters. That's why they're called filterable agents. All right, now aside from that, they are also considered obligate intracellular parasites. Now take note, I remember sa una, nagsugod pa lang pandemic, akong gi-clarify ni sa akong mga public health students kay some posts have been describing viruses as parasites, no. So kani siya na line dears, no, I hope you don't get confused. Your virus in itself, it's not a parasite per se. When we mean parasite as the parasite that we know from parasitology, like the worms, the protozoa, they are not the same biological organism. Okay? Your parasites and viruses are different in terms of biological organisms or biological composition, in terms of their genome and all that. Okay? So, muna siya difference niya. However, your viruses are considered parasites in the context that these viruses require hosts to live and to survive. Alright? So dito siya mo differ, okay? Or dito siya muna pasabot sa description ng parasites ang mga viruses. So I hope dira mo ma-confuse, okay? These parasites, or describe ang viruses as parasites in the sense that your viruses require hosts for them to survive and to live. Because again, di ba, what do we describe a parasite as, di ba? Parasites are organisms that require hosts for them to live. Okay, and that is your virus. Okay, so ang description lang na parasite dira dears, gi-describe ra siya in the context that they require a host for living. But in terms of biological composition, in terms of genome, in terms of characteristics, they differ from the parasites that we know, such as atong na-discuss gyud sa parasitology, like the worms, no, the protozoa, inana. So they are really different from the viruses. Okay, so I hope na gets lang. All right, so obligate intracellular, nasa pangalan na, obligate, kailangan yun, no, need yun, necessary yun na naa sila sa sulod sa cell. All right, because without cells, no, your viruses will not live. Okay, because they require to be inside the cells because they need the cells for them to survive. All right, so I hope na gets ra nganong gitawag siyang parasites pod. Okay, all right, because again they depend on the biochemical machinery of the cells for their reproduction and for their survival. All right, now reproduction of viruses, they differ also from your bacteria and parasites, no. They reproduce by assembly of components, not by binary fission, unlike your bacteria and some of your parasites. Now, another one is your viruses. They are very type specific, no? They can only infect a particular type of tissue, a particular type of cell, and this is known as your tropism. All right, so not all of your viruses will infect a lot of cells. No, they have again specificity. They can only infect a particular type of tissue, a particular type of cell, and that is known again as tropism. Kani siya na characteristic. So na yung mga viruses na predilection or ganahan sa mga cells sa lungs, uban na ganahan sa heart, no, uban na ganahan sa imuhang intestines and all that. So depende sa ilahang tropism, okay? But napay ubang mga viruses that they can infect a wide range of cells because again, they have receptors, or they can infect, or they have receptors to to different types of cells. Okay? All right. Now next, viruses lack again the capacity protein synthesis and the rest, no. So they really rely on the cell, they really rely on the host for their survival, for the production of their necessities in terms of their structures, their proteins, and all that. Okay, all right. Now again, take note, the genome is either, please take note, Take note of this. It's either RNA or DNA, not both. Okay, please take note talaga. Sinasabi ko talaga. Very important because there are questions in the boards na inaani na like, which of the following is a characteristic of a virus? Which of the following is blah, blah, blah. So please take note. Their genome can only be one. Okay, either RNA or DNA and never both. Okay, that's another unique characteristic of viruses. They don't have RNA and DNA at the same time. Unlike your unlike your parasites, unlike your bacteria, all right? Unlike humans, of course, we have both RNA and DNA at the same time. But for your viruses, they only have one. Okay, either RNA or DNA. Please take note ha, either. Okay, but never both. Please take note. So loyal sila. Either RNA or DNA lang. Di para sa uban na magdungan ang duha. Charot. Okay, ayan. So, either DNA or RNA lang. Alright. Now, classification of viruses is usually based on the nucleic acid type. Example, the RNA viruses or DNA viruses. Another means is for the size and shape of virion. Is it big ba in terms of their size? And the presence or absence of an envelope. Yes. So, those that are enveloped and those that are naked. Alright. So, muna siya itong mga description nila. Alright. Now, for pandemic, of course, this term is very common na. Common na in this time and in time and age, of course, with this COVID-19 pandemic. So the pandemic refers to the emergence again of a new viral disease across a very large geographical region. So usually it involves the world, no? If it involves only the country or mga little places lang or mga pila ra ka countries or within the country lang, that's known as an epidemic, okay? Pero kung pandemic, it's really affecting the whole world na yun. Alright, okay. Now, there are some terms known as your antigenic drift. Ayan. So, when you say antigenic drift, these are only minor changes that occur infrequently. So, from time to time, ang viruses, na sila changes in terms of their structure, in terms of their virulence, and all that. But these are usually not significant kaayo to the point na makakause siya og mga severe symptoms or severe diseases. These are known as antigenic drift. So, minor lang, minor. Ang antigenic shift, these are major changes yun that result in the novel mga viruses. And this is usually common sa mga influenza na viruses, no? Especially influenza type A, because these viruses exhibit antigenic shift in the way na ma-change gud iyang entire structure, ma-change gud iyang mga virulence to the point na naibagong strain na pod, no? So, inana siya na intense, no? Antigenic shift. Okay? So, please take note, kung drift ra gani, minor changes, all right? Kung antigenic shift, major changes. So, how do you remember? Kung mo shift ka, di ba? Example, mo shift ka course, that's a major change. That's a major decision to make, no, in your college life. So, major changes, major decision, so that's antigenic shift. Okay. All right. Sige. Next, you have, again, antigenic drift, minor changes that occur infrequently. All right. Okay, sige. So that's for the introduction for the viruses. Now, we also have what we call the viroids or viroids. These are virus-like particles, but they're not really viruses, no? These are just acellular, naked lang sila. Usually, they only contain the genome in itself, no? Example, here's a picture. Pwede siyang single, stranded, or branched. Okay, so genome ra gyud siya, RNA, usually RNA ra, and then muna na siya. And it can infect organisms, usually sa plants. Okay, sa humans, we have Delta virus, no, your HDV. Yeah, tama ba? Your Delta virus, yeah. Delta virus is a viroid or viroid. It's a defective virus, meaning dili siya complete, no? Wala siya outer covering and all that, just the genome in itself, but it can cause infection. But majority of viroids, they affect your plants. Okay. All right. So that's for your viroids. All right. So we go now to your viral structure. No. So what is the components of your virus? No. So your viral structure, again, the entire infectious particle of your virus or the mature, ayan, mature, complete yes, virus particle that is known as your virion. Okay, the virion is the entire complete infectious particle of the virus, and these are composed of three parts. Of course, the inner genome or the nucleic acid core, which can again either be DNA or RNA. Next, of course, there's a protein coat that surrounds the genome, and that is known as your capsid. Okay? And in some of the larger viruses, not all ha, dili tanang viruses na ay envelope. Alright? So only some, especially those that are large, big na mga viruses, they have what we call an envelope. So nasa pangalan na envelope, it's an outer covering. Okay? So three basic no or major structures good. You have the genome, the nucleic acid core, which can again either be DNA or RNA, all right, nasa tunga, and that is covered, outer covering by again a capsid, which is made of protein, and then some viruses lang have the outermost covering pagyod, which is your envelope. Again, not all of your viruses have the envelope. Okay, please take note of that. All right, so that's for the initial of your viral structure. Okay, all right. Okay. Sige, sige. All right. Now we go now to the viral structure, no? Components, all right? Starting first with the capsid. So for the capsid, this is just the protein coat, no? So this is the coat that is covering you the nucleic acid core, okay? Which could either be RNA or DNA. Now sa capsid palang daan, daghan ng different types, no? Daghan ng possible different configurations, no? Na yung ibang viruses na helical, by the name itself helical, ayan, twisted, no? Twisted iyahang capsid. Another one is icosahedral, meaning ni confer siya og shape, medyo cubical iyahang shape and siya 20 ka flat sides, all right? If mo siya lang taon 3D, ayan, so na siya mga flat sides, so icosahedral. And lastly, complex, no? An example that is pox virus, meaning dili siya helical, dili po siya icosahedral, lahi siyang shape, and usahay medyo brick-like iyang formation or different shapes siya, okay? So that's complex. All right. Now, the individual protein, ayan, protein components of your capsid is known as the capsomer, okay? So individual protein subunits of your capsid is known as the capsomer. And together, kani sila, we're high five, charot. Together, kani sila, they are known as the nucleocapsid. Alright, so ito na siyang i-draw para mas mag-gets. Alright, sana makita naman. Alright, so sa tunga, of course, is the nucleic acid core. Maklaro na ba? Ayan. And then, of course, the outer covering, sulod na niya, is, ato na triangle ha, sorry, Illuminati confirm, charot, is your capsid. Okay? So, kanarang outer covering is known as the capsid. Okay? Now, the inner is your nucleic acid core. Together, kanisla together, alright, this is known as the nucleocapsid. Okay? Alright, nucleocapsid. Alright? So, I hope na-get siya to siya. Okay. So, ayan. Capsid, if katulad yung outer covering, nucleocapsid together now with the nucleic acid core. Okay? Alright, ayan, sige. That's for the initial structure of your virus. So, here's a picture. Ito na lang i-present. Ay, para mas maklaro ko i-zoom. Okay, ayan, sige. Ayan, so naked icosahedral, as you can see, icosahedral, mura siya cube, no? Cube-like, ayan. So naked siya, meaning wala siya outer envelope. Ayan. Next is your helical. As you can see, naked gyapon pero helical ang itsura siyang capsid. So, mura siyang spiral. And this one, icosahedral pero na siya outer envelope. So, enveloped icosahedral. And then lastly, of enveloped helical. As you can see, na siya envelope, outermost pag yun. And then you have kaning black is the genome. And then kaning surrounding na mga circles, that is the capsid. Helical ang shape kay spiral siya. Alright, so pwedeng enveloped or naked imuhang virus. Okay? Alright, ayan. So that's for the structures. No? So capsid is the protein coat. Ayan. Protein coat that surrounds your genetical or genome, your genome. And then the individual proteins that make up the capsid, that is your capsomer. Alright? And again, together, the capsid plus the nucleic acid genome, that is known as your nucleocapsid. Alright, okay. Next, you have the envelope. Again, some viruses only have the envelope. It's not all viruses na envelope. Alright? So, again, once the virus is non-enveloped, we call the virus as naked. Okay? So, naked, they don't have the envelope. Alright? So, and usually the naked viruses, they are the more resistant, no, to environmental changes, to detergents, no, to acid, pH, and all that. And usually, kani mga naked na mga viruses, sila usually ang ma-transmit fecal orally because again, they can withstand the changes and the acidic environment in the gastrointestinal tract, the naked viruses. In contrast, your envelope viruses, as you can see, it's covered, no, with an outermost covering. And usually within the envelope na yung mga glycoproteins, all right, mga spikes, ayan, that we use, the term that we use, spikes, and also other mga structures that the virus use as a means of attachment, usually to the host or to the to the cell that iyang gina-infect. Okay? So, nasa surface sa iyang envelope. Alright, so, but again, usually, if envelope viruses, usually sila ang mas prone to changes in the environment. Okay? Especially to detergents, mamatay na sila sa mga changes in temperature, pH, and usually, since they are very much sensitive, kani sila ang mga viruses na dili ma-transmit through fecal oral. Okay, usually they can be transmitted through droplets, no? They need to be in a wet environment all the time. That's why they can be transmitted through fluids, no? Like saliva, semen, no? And all that. So usually kato mga envelope viruses, sila ng mga sexually transmitted diseases or sa respiratory ma-transmit. Example, your coronavirus, no? Your coronavirus is enveloped, okay? And the envelope, as I've mentioned, again, is very sensitive to temperature changes and also to acidic environment, to detergents, and all that. That's why they need to be in a wet environment, aqueous solution usually, or basta wet environment para mas makasurvive sila. Alright? Now, this envelope, usually, it comes from the host cell. Pag release, no, during viral application sa imuhang virus, kani siya will iyahang dalon pod og apil ang cell membrane sa imuhang cell or sa host na kung asa siya nag-infect. And this will now serve as the envelope. Now the envelope man good is composed of, 'di ba, cholesterol, 'di ba? Remember mga phospholipids and all that. So therefore, phospholipids, they are polar, all right? So therefore, mas easy silang ma-destroy by polar na mga solvents such as your alcohol, your detergents. So, mas easy silang ma-disrupt. So, pasalamat ito ng coronavirus sa COVID-19, yes, kay enveloped. So, mas easy lang siya matay sa atong pag-disinfect, sa atong kamot, no? Using alcohol-based cleansers and soap and water put pag-wash. Because again, the envelope is polar. Kaya that's made of phospholipid and all. Kaya gikan man na siya sa cell membrane pod sa iyahanggi na infect na cell, di ba? So, therefore, polar na siya, like charges attract, so therefore mas easy lang siyang mamatay sa atong mga alcohol-based cleansers and disinfection purposes, di ba? Or mga disinfection methods na to. Ayan. So as you can see, ang naked is much more resistant. As you can see, di ba? Basically, ang imuhang naked viruses, nucleocapsid ra gyud siya, di ba? So you can see you have the genome and then you have the capsid, di ba? As we have mentioned, that's the nucleocapsid. So therefore, your naked viruses sometimes are also called the capsid viruses because again, they don't have the outer envelope. So basically, it's a nucleocapsid lang yun, all right? And this capsid, di ba, is made of protein, and proteins are very very much resistant, no? And very much strong in terms of exposure to mga detergents and all that. So medyo lisod lang patch yun. Compared to your your envelope viruses, actually, it's the nucleocapsid, giadano nimo siya outer na envelope. Alright? So that's the difference, diba? Same as like structure. Ang difference lang yun is the envelope found in your enveloped viruses. Okay. And as I've mentioned in the envelope, alright, envelope ng mga viruses, on the envelope itself, you can see again glycoprotein spikes, alright? That is used for attachment purposes, alright? There could also be mga other viral structures, no? Usually used for attachment purposes, good, to the for infecting other cells. Okay? Usually, ang imong naked viruses po, they may also have mga glycoprotein spikes or mga proteins sa ilahang surface, but majority makitan gyud siya sa enveloped na mga viruses. Okay? Alright, so that's for the envelope. Again, not all. Okay? Not all of your viruses have the envelope. Only some. Okay, alright. And usually, again, kung envelope siya, mas prone siya, mas sensitive siya to disinfection, detergents, environmental changes, and all that. Okay, alright. Now, other structures, you have the matrix proteins. These line the inside of the envelope and is usually used for assembly purposes of the virion or mga baby na viruses na gina-produce. Alright. The tegument is the space between the nucleocapsid and the envelope. And in the tegument, you can see other enzymes, proteins that are necessary for viral production and viral synthesis. Okay, so pwedeng mga matrix proteins makitaan po diri sa imuhang tegument. Okay? Ayan. So, that's for the viral structure, no, the basic viral structure. Just take note lang yun, inner nucleic acid core. It could be either, ako na gina-emphasize, either RNA or DNA. And then again, covering that is known as the capsid, okay, which is made of protein. Each subunit is known as the capsomere, which makes up the entire capsid, all right? And the nucleic acid genome plus the capsid, that's all together described as your nucleocapsid. Okay, alright, ayan, sige. Okay, now if naked ang virus, again, wa siya envelope. It's just like the nucleocapsid in itself. Pero if envelope siya na siya outer covering pagyud, outermost covering, and usually, kaning envelope, it comes from the cell membrane of the host, no? Paggawas sa imuhang new virions na produce, iyang i-acquire ang imuhang envelope, no, or cell membrane from your host cell. All right, and that becomes again the virus's envelope. And again, envelope viruses are much more prone, much more sensitive to detergents, changes in the pH, and all that. Okay, all right, ayan, sige. That's for viral structure. And again, here are the summary of the types of viruses. So again, helical na mga viruses. Again, the capsid is helical. The genome is also helical. So an example of that is tobacco mosaic virus. This is not a human pathogen. It only affects usually mga plants. Next, you have the polyhedral. Ayan, as you can see, 3D shape na siya marag cube-like appearance, no? So example na adenoviruses. Spherical viruses, you have usually the enveloped, all right? Usually mga coronavirus. And you have the complex viruses, which is usually seen sa mga bacteriophages. So when we say bacteriophages, these are viruses that infect bacteria. Alright? So, mara siya og kanang spaceship, no? Actually, it's described as parang, eto, nandito, lunar lander. Ayan, mara siya kanamulan sa moon na mga ginabuhat sa NASA and all that, diba? So, that's a bacteriophage. It's a virus that targets a bacteria. Alright, okay. So, those are the summary for the types of viruses. Alright, okay. Alright, okay. So, we continue with your viral replication. Alright, so part 2 na ni sa video. Basin ako rin siyang chop siya 20, ano dagay siguro, no? 20 minute or 30 minute, 40 minutes siguro. Sige, we'll see. Alright, sige lang. Alright, so we'll go now to viral replication. So by the name itself, we'll look into the how viruses reproduce, no, in terms of when it comes to the body and all that. Now for viral replication, again, as we have mentioned, we have established already that the viruses, they are intracellular, they are strict, obligate yun, kailangan yun sila cells, kailangan yun sila nasa sa sulod sa cells, because if wala sila dito, then they cannot survive, they cannot live po, and they cannot replicate. Okay, because they need the cells to survive, no? How do I live without you? Ganong level. All right? So they need the cells, no? For them to survive, for them to replicate. Because it's through the cells, no? Ilang gina-hijack ang cells, ginagamit nila ang cells. Ginagamit nila ang biochemical, ang ilahang mga enzymes and mga procedures, mga processes within the cells para makaproduce sila og new na mga viruses. So ilang gina-hijack, actually gina-hijack nila ang biochemical and all their mga nutrients sa cells na ilang gina-infect para makaproduce sila bago na mga virions. So, ina na sila katikasan. No? Grabe yun. Okay? So, manggagamit, user. Okay? Dili sila user-friendly. Kaya di man sila friends. Pero, user. So, may mga kilala kayong ganyan. Charot. Okay? Ayan. So, again, they are parasites. No? In the context lang that they require host, they require cells for them to live. Okay? But again, in terms of biological composition, in terms of characteristics, again, they are different from the parasites that we know, such as your worms and protozoa. Okay, so again, ako na na-emphasize, but I hope na gets wrong difference. Alright, okay, again, the cell will act as a factory. Again, ang cell, ang ga-provide gud og nutrients, og mga machineries, no, mga enzymes, para lang gyud sa virus, no, para gyud makaproduce og new virus. So basically, the virus, again, hijacks the cells na ilang gina-invade. Alright, now for the viral replication cycle, it can be divided into different phases. Number 1 is, of course, the early phase. So by the name itself, it's the early phase, meaning this is the start pa lang of something new. Charot. This is the start of infection. Diri na time, bago pa lang ni sulod ang virus sa imuhang cell. Alright? And then afterwards, nag-produce na siya og after attaching, no, or after penetrating the cell, of course, iyahang gina-uncoat, tangtangon ni iyahang capsid para ma-expose ang genome, and then the genome will then be replicated inside the cell to produce more viruses. So that's the early phase. All right. Now the late phase is the start of genome replication and macromolecular synthesis, meaning the production of viral proteins and other structures of the viruses that they need para makasurvive sila or para makaproduce sila new na mga virions or viruses. All right. Now the eclipse period, this is the time in which walay ma-detect, no, na mga viruses imuhang samples. So eclipse, no, wala kayo nakita. All right. So it's the period of uh, non-detection. Alright, so wala siya nakita. Alright, and this eclipse period is found in the latent phase. Ayan, so the latent phase, ang makitaan dira is the eclipse period. So nasa pangalan na latent phase, again, this is the period in which wala pa nakita ng mga viruses from the sample. Because again, it's still, the viruses are still within the cells. Nagpadayon Japon sila og replicate. Nagpadayon sila og produce og mga macromolecular requirements, mga proteins and all that. And then of course the burst size, this is now the release, no, the amount of yield per cell of mga infectious na virions or infectious na virus. So this is the number of particles na ma-produce per cell. So this is known as the burst size. Alright. Now, each infected cell, again, may produce as many as 100,000 particles. But about 1 to 10% of these particles may be mutated, no? Or maybe, only 1 to 10%, sorry, ang infectious. So, bahalag 1 to 10% pa rin. If you think about it, if you mo gamito ng math, ang 10% is, Ayan, medyo, nagloading ko light sa math. 10% mga 10,000. Am I right? Am I right? Oh my God, Mark. Yeah, I think. So 10,000 pa rin na infectious virions or viruses are still a significant number. Bahalag dili gyud 100% of the 100,000 ang infectious. Those 10,000 particles na infectious, meaning complete gyud iyang structure, no? Walang bahid ng mali. Wow. Perfect yun siya. Complete infectious virion. Bahalag 10,000 na sila, kay 10% raman, no? 1 to 10% of the 100,000 na ma-produce, dako ra gyapon siya na amount. It can still infect, it can still establish a new infection on another host or pwedeng another cell, diba? So, bahalag unsa kagamay. Alright. Now, the remaining, of course, na percent na na-produce, of course, these are those that are mutated, no? Those that with mga errors in terms of mutation, in terms of the products that they produce and all that, no? So, these mutants, again, or mga mutations po, they can also, no, facilitate new infections, and also can cause either more severe ba or less severe na type of infection. So, nagyapoy advantage or nagyapoy murag edge, okay, or nagyapon siya dulot, nagyapoy effect ang imuhang mga mutations. Bahalag mutated sila or sayop sila pagkahimo. Ganun, okay? Alright, so that's for the phases of viral replication. So, early, of course, late, and then the latent phase, which again consists of the eclipse period, and then finally the release, or again the burst size. Again, the burst size is the number of virions na produce per cell. Okay, alright, ayan. Now we go na to the picture. Ayan, as you can see, in the start of the infection, of course, mugamay siya, meaning kaning detectable na virus concentration. Because again, they are well within the cell, still replicating, still producing the protein. So, di sila ma-detect. Wala pa'y infectious virion na ma-detect sa imong samples. But as you can see, pag-abot sa late, nagsugod nag-increase because again, padulong na dira or naghinahinay nag-release o mga new virions from the cell nag-infect. And then, of course, the burst size mo plateau na. Kaya dito na nagkadaghan, constant na ang production of your new virions from the cell. Okay, so as you can see, ayan, plateau na siya because constant na lang ang production. Okay, constant ng production, not really plateau, pero pasaka pa siya, no? Constant ng production of new virions from the cell. Okay, alright. So that's for the phases of viral replication. Alright, but we go now to the steps, no? The steps in viral replication. Meaning, katulad yung mga like unsa joy mga steps nagloading pa ko lights mga procedure na gi-follow gyud sa viruses in infecting new cells or mga host cells. Alright, so we'll start first with attachment and penetration. Ayan, so by the name itself, no, as we have mentioned, in the outer surface of your viruses, they have viral structures usually made of proteins that they use for attachment. Okay, because how will they establish an infection or how will they infect a cell if dili sila musulod sa cell? So before they go inside the cell, they have to first be attached to the cell na ilahang gina-infect. And that is the process of attachment. No? So, again, the virus, they have their own structures on the surface that will interact, that will attract, or that will recognize, ayan, a suitable host cell and a specific receptor na naa sa imuhang host cell mismo. So they will interact with each other, alright? And pag interact nila to each other, it will now proceed to penetration, okay? Because what happens, magud, is the host cell will recognize the virus na murag, okay, this is something that we need, or this is something that kailangan nato ipasulod. Ma-recognize siya because of the receptor, no? The receptor will bind to each other on the surface of the host cell and on the surface pod a different receptor sa imuhang virus na structure sa outer surface. Mag-bind sila together, of course, so mag-abot sila or mag-abot sila og understanding na murag yes, okay, we know each other. Ayan, so masulod na ang cell through penetration, okay? And these structures, again, on the surface of your viruses, they are known as the viral attachment proteins, VAPs, or also known as adhesion molecules. Ayan, so VAPs, no? Viral attachment proteins, no? From the top, make it drop. That's a VAP. Charot. Okay, charot. Ayan, sige. So, VAPs, again, structures on the surface of the virion, na capsid or envelope, that may bind to your cell receptors. Okay? And again, they could be proteins or carbohydrates found on the glycoproteins or glycolipids of your host cells. Alright? So, the viral attachment proteins, here's an example. The virus, and then unsa ni mga viral attachment protein niya. Meaning, these are structures, again, found on the surface of the virion. Okay, so sa virus ni siya na structure known as your viral attachment proteins or adhesion molecules. So, ayan, memorize naman, no? Di naman kayo lumalabas sa boards, pero take note lang, very important, HIV. Ayan. So, unsa ka HIV, GP120, glycoprotein 120. Nako, very, press the buzzer, agad-agad. I feel like na-discuss niya yung IS. Hopefully, na-discuss niya sa IS, HIV serology. Yes naman. GP120, one of the most important markers then for HIV. Alright. And influenza A, of course, the HA glycoprotein or hemagglutinin. Ayan, very important. So, later when we go to the different families of viruses, ato na po ni siyang i-discuss. Charot. Okay. Now, for the virus, that's your viral attachment proteins. Now, on host cells or on human cells, this is known as the viral receptors, no? So, for CMV, ang iyang ginagamit is the integrin na receptors ato mga cells. Your EBV, of course, another very important. I hope na discuss po ni sa inyohang IS lab, EBV serology. Unsa gina-infect sa mong EBV, of course, the B cells because they have the receptors na kailangan sa imong EBV, which is your CD21. Oh, nako talaga. Press the buzzer na good ni. Oh my God. Again, receptor needed by your EBV is your CD21. And of course, HIV, wag na maisip, of course, the CD4. It can also use the chemokine receptors CXCR4 and CCR5. You also have parvovirus B19, the P antigen, of course, found on RBCs. And then you also have the rhinovirus, rabies virus, nicotinic acetylcholine receptor, which is found in neurons. That's why usually sa brain ang gina-infect sa mga rabies. And of course, influenza A, sialic acid, usually found in your respiratory tract and other cells po. Alright, pero very important na yun, EBV, nako. Review na yun ni siya, dears, no? CD21, EBV, of course, HIV, CD4, di na mawala. Okay? Ayan. So, that's for the receptors. Kani sila again na table, kaning sa lower left, kani siya. These are the receptors found in the cells nato, sa humans, that can be used, or muna siya ginagamit na receptor sa imuhang viruses. Kani siyang sa taas, these are the attachment proteins na naa sa ilahang but nasa virus itself, nasa surface sa virus. Okay? Alright, ayan. So, that's for the first step. Attachment and penetration. Of course, unsa nila pagsulod sa cell, if sa sugod pa lang, di sila maka-attach or di sila tapot sa ihang ganahang ma-infect na cell. Alright? So, that's for attachment and penetration. The first step of viral replication. Of course, next, is of course, the uncoating. Meaning, pagsulod na sa virus sa imuhang cell, of course, okay na. The virus is happy. Kaya yes, nakasulod ako sa cell. Okay. Alright, so, first step done. Pak, ganun. So, ang next akong buhaton na sa virus is tangtangon na ako ang capsid. Because again, my main goal is to produce more virions or to produce more viruses. Okay? So, how will I do that? It's through the replication of my genome. Okay? Ako pang padaghanon akong genome para mas daghang viruses or virions na ma-produce. Alright, so uncoating, nagtangon niyang capsid, no? Iyahang hubuon, uncoating, nasa pangalan na, no? And depende siya kung DNA virus, of course, it will integrate into the nucleus. Kay DNA good mismo. So DNA man ang virus, musulod siya sa ato ang nucleus, ato ang cell, because humans as we are, di ba? Ato ang cells contain DNA. So, dito siya musulod sa nucleus. Whereas for the RNA viruses, they are already RNA. So, sa cytoplasm na lang sila mag-replicate and all that. Okay? So, example lang for pox viruses. Because for pox viruses, they will uncoat in the cytoplasm. Alright? Okay. Now, after that is, of course, macromolecular synthesis. So, nasa pangalan na, uncoating, no? Uncoating, ang procedures uncoating or ang purpose is to express the genomic component of your virus. So after which, of course, is macromolecular synthesis. Meaning, i-replicate na ang genome sa mong virus, mag-produce na sa viral proteins and other structures. Alright? So again, all DNA viruses, sa pangalan na, the replication or macromolecular synthesis happens inside the nucleus, except lang for pox viruses, alright, because sa cytoplasm siya, alright? And for all RNA viruses, they replicate in the cytoplasm except kinsaman, influenza viruses and retroviruses because they replicate within the nucleus. So take note lang kung kinsa itong mga exceptions, alright? Because your retroviruses, di ba, they have the reverse transcriptase enzyme. So ang buhaton sa retrovirus, iyahang i-convert yung RNA to DNA. So therefore, musulod siya sa nucleus para dito siya ma-replicate. Alright. And of course, very important na ma-produce ay muhang macromolecular synthesis are the different proteins. Ayan. Different proteins, different structures, enzymes needed by the virion. Alright. And that is your viral protein synthesis. So it will depend on the host cell ribosomes, transfer RNA, and of course, mechanisms for post-translational modification to produce their proteins. Because again, muna kailangan yun sa viruses, no? They need the proteins para establish sila or maka-create sila og new na mga virions. Ayan. Alright, sige. So, here's an example. Hala, asa to? OMG. Okay. Alright, ayan, sige. So, that's for the macromolecular synthesis. Okay. Okay, so after the macromolecular synthesis na part, okay, so atong lang tawon ning murag summary, no? So kanina siya na part dears, no? Dili lang kay kumu in depth na po. Kay medyo nalimot ako sa akong biochem lunch, no, in terms of mga replication. But again, take note lang, 'di ba? From DNA, 'di ba? I-replicate ang DNA, all right? And of course, from DNA, transcription happens also, alright? And transcription will produce mRNA, diba? That's the central dogma. And mRNA will be translated through translation to produce the proteins, alright? So DNA, transcription, mRNA, and then mRNA translation, mahimo siyang protein. So mura po na gina-follow sa imuhang virus. Alright, now take note, diba, na yung mga terms like double-stranded, single-stranded. So, we'll look first at double-stranded DNA. So, double-stranded DNA, we have the polyoma, papilloma, adeno, herpes, and pox na mga viruses. So, double-stranded, by the name itself, no, double-stranded, that's following the normal DNA structure, especially sa humans. So, after dsDNA, again, follow siya sa normal procedure, ma-transcribe siya, mahimong mRNA, and then from mRNA translated to protein. So, inana po ng mahitabo anin sa double-stranded DNA. So, yahang DNA, alright, will be transcribed to be mRNA, and then mRNA from there will be translated to the different proteins. So, ano siya mga proteins are the different viral proteins na. Alright. Now, si Parvoviridae, ayan, siya lang different, single-stranded siya. So, what happens is a single-stranded DNA, i-convert sa siya or gaman sa siya another strand of DNA to become double-stranded DNA. So, from single-stranded, may mo siyang double-stranded. Okay? And then, the procedure then follows the normal procedure. Again, from dsDNA, mahimo siyang mRNA, and then finally, the proteins of the virus. Now, for positive sense RNA, when you say positive sense RNA, it already follows the same structure with your mRNA. So, kung positive sense RNA siya, mRNA na siya in the structure. So, madiretso na lang siyang mahimong protein. Okay? And ang ma-produce na protein is known as a polyprotein, meaning taas kayo siya na protein or one structure agad siya. So, what happens is i-cleave niya siya into the different types or into the different smaller proteins na need sa imuhang virus. Basta ang point lang kung positive sense siya or positive stranded RNA, similar structure na yung RNA to the mRNA. So, diretso na siya. Pag sulod niya sa cell, na host cell, kato yung mRNA, diretso dayon o translate or diretso dayon siyang i-translation para mahimong protein. Okay? Now, for the negative strand RNA, ayan, the negative strand RNA, it's like a template muna siya para mahimong mRNA. So, same ra gyapon, gamaan siya og mga different mRNA, different mRNA na mga strands na ginagmay. So basically like kanina portion kanisa ang igam-an na mRNA, kanina part, then basta ginagmay siya ng mga parts sa imuhang negative sense RNA. And then of course, may mo siyang mRNA, messenger RNA, and then becomes the individual proteins. Now for double-stranded RNA, which is found in your Rio Viridae, double-stranded RNA ra gyapon siya. Alright, what happens is of course, it will be translated to mRNA. Okay, himay mo siyang i-chop off siya. Alright, and then para mahimo siyang mRNA, parehas ani. And then, na mga individual proteins na ma-produce. Okay? Alright, so that's your double-stranded RNA. And of course, for retroviridae, so RNA siya gikan, pero naman siya reverse transcriptase na enzyme, ma-convert ang imong RNA sa imuhang virus to the double-stranded DNA. And this double-stranded DNA will then be incorporated into the hosts, into the cell's genome, and then and then follow the same procedure gyapon, normal procedure sa imong double-stranded DNA. It will be transcribed to mRNA, and then mRNA will be translated to the different proteins. Okay, so that's the summary lang of the different genomic, na parang genomic processes of your viruses depending on the type of genome that they have. Okay, all right, so that's for that's for their genome. Alright? That's for macromolecular synthesis. So, nasa pangalan na, macromolecular synthesis, the main point is it's for production of the necessary proteins, necessary structures that your virus needs. Okay. Alright. Now, of course, after producing the necessary proteins na kailangan, of course, we now proceed to assembly. So, nasa pangalan na, assembly. Okay? So, assembly is basically dili na sa katong SUSG, charot. Assembly is basically nagkumpul-kumpul or nag-compile, nag-compile sila or nag-build small na mga proteins until to the larger ones. So, nagkumpul-kumpul or nag-compile sila. That's assembly. Alright? And then, each part of their virion, they have recognition structures. So, nasa mga recognition structures na, okay, need na ko ni siya na protein, kanito siya kung need na nucleic acid, etc., etc. And mga interactions between these structures para ma-assemble sila into the final structure. It's very amazing, actually, to think about, no? And then, the site and mechanism of your assembly happens also at the same site kung asa nahitabo ang replication. So, for DNA viruses, it happens in your nucleus, and for RNA virus and pox virus sa cytoplasm. And errors, of course, as we have mentioned, are made during the assembly. Alright? Because again, dili malikayan, na yung mga errors, of course, and this will result to mga mutants, na mga viruses. Alright, okay, so that's for assembly. And of course, the last is your release. So after kanang building, after building the different and after getting the different necessary structures of the virus, of course, they then produce to the release, para mo infect og new cells. So release, magpagawas. Release, production. So release, lysis of your host cell, like mo burst yun siya, that is seen in mga naked na viruses. Alright? So, mamatay yun ang cell, ang host cell, kay ilahang i-erupt, ilang i-burst ang cell. In contrast, imo mga enveloped viruses, they release through budding. Alright? Meaning, pag bud lang, ilahang kanina picture, ayan. Gamito na nila, gamit sila sa cell membrane, paggawas nila, okay, ayan. So, paggawas nila, magdala sila og cell membrane, okay, with them, and that becomes the envelope, ayan. And they don't kill the cells in this process. So, that's another advantage because the cells are still alive, so makaproduce pa siya more na virus, di ba? Continuous ra gyapon ang production sa mga new virions because the original cell na iyahang gina-infect, wala ra namatay. So, padayon og Japon og synthesize and all that. So, mas daghang virions na pwedeng ma-produce pa continuously. Alright. And again, the virion is known as the complete virus particle. Again, complete virus particle together with the envelope, if envelope mang virus, with the capsid and then the genome and the necessary proteins inside para maka-infect. Ayan. And also, kung naked siya, nucleocapsid, the nucleocapsid and the necessary proteins and enzymes na kailangan para maka-infect siya. Ayan, that's virion. Okay, the complete infectious virus particle. The size is about 0.01 to 0.3 micrometers. Okay, ayan, sige, that's for the release. Okay. Now, ayan, nasa picture na, di ba? So, it starts with attachment, okay? And then penetration, meaning nakasulod yun siya. After penetration, of course, happy na siya, kaya na siya sulod sa cell na yung gina-infect. Okay? Tatangaw na niyayahang covering, of course. Maghubo yun siya ba? Ayan. Uncoating, all right? And yan ang express iyahang genome, all right? And this genome will be incorporated into the natural and normal processes of the host cell. Okay, and then of course, ma-produce na mga necessary proteins na kailangan sa imuhang new virions, and these new proteins will then accumulate, okay, and they will build each other or mag-build na og new na mga virions, ayan, and they will now be released, no? It's either lysis para sa naked, ayan, which bursts the whole host cell, or kung envelope siya, it's through budding, alright? So, mugamit siya og cell membrane sa imong host cell. Paggawas niya, of course, dili ra mamatay ang cell. Kay cell membrane naman yung gikuha. So, pagtangtang ani, mabalik raman Japon or replenish ito mga phospholipids niyang gikuha as the envelope. Alright? So, that's for your, for viral replication na steps. And we have a mnemonic for that, and that is APUMAR. Ayan, diba? So, APUMAR, para mas easy ang life, APUMAR. Attachment, penetration, uncoating, macromolecular synthesis, assembly, and then finally, release. Okay? APUMAR. Ayan. APUMAR. Okay? Alright. And if you want to, i-relate siya sa real life, o, diba? If makamit kag someone, nimong bet, so ma-attach ka. Alright? So, after nimong attachment, that's say next, penetration. No, penetration in terms of getting to know, penetrating your barriers, your walls, okay, that you have set high to protect yourself from hurting and all that. So, penetration. Nawala na, natangtang na tanang walls. So, penetration. After that, uncoating. Maghubo na mo. Ang basta. Uncoating, meaning, you became more vulnerable with each other. Ayan. You then get to know each other's demons, your strengths and weaknesses, no, your love language, ganun sa love life. And then after, of course, is macromolecular synthesis. So you make the relationship more stronger. No, you build the relationship, you nurture, you produce more proteins, you produce more, you strengthen the relationship. Ayan, macromolecular synthesis. And then assembly. Ayan, so what you have produced, okay, what you have nurtured, yun yung ipalago, assembly, ayan, or assembly, or in the long run, assembly, mag-marry mo, no? Ayan, magpakasal mo, assembly. So you build a family, pack ganun. And then lastly, release, and that is now the production of your offsprings, mga children. Nag-release na mo children, nag-release na mo mga new persons in the world. O, diba? So pwede na to siya ma-relate sa atong love life, atong mga relationship. Okay? APUMAR. Attachment, penetration, uncoating, macromolecular synthesis, assembly, and then finally release. Ako kay Kutob Radyo Kog, penetration. Charot. Wala pa ko naabot sa macromolecular synthesis. Mga Kutob ra kog, uncoating. Sayang. And then after uncoating, ghosting na. Joke na. Hurting ng next. Charot lang. Okay? So again, atong goal dapat maabot sa release na part. Okay? Sa ato ang mga relationships, release, okay? Dapat produce tag mga own children. Pero again, of course, really required yun naman. Pero ayan, just to, relate, no, to real life. APUMAR, attachment, penetration, uncoating, macromolecular synthesis, assembly, and then finally, release, no? So, in summary yun. Napaginay siya yung mga small steps in between, pero gi-generalize na to, APUMAR, para easy ang life, di na tayo mag-complicate sa atong life. Okay? Alright, ayan, sige. So, I hope na-gets ra to siya, and I hope effective lang po to ato ang mnemonics. Okay? So that's the viral replication. Meaning, muna buhaton jud sa virus sa kada cell na ilang gina-infect. Okay? Ayan. So, start with attachment, penetration. The end goal is release. To produce more virions, to produce more viruses na maka-infect og new host cells or establish infection ba into other cells. Alright? Muna dyan ay goal sa mong virus dears, no? So, goal sa viruses regular is to survive, no? Actually, sa tanang pathogens, ilang goal regular is to survive. Actually, I feel like they don't know that they are causing diseases, no? Actually, ang ilahang goal regular is to survive, no? Whatever happens and whatever unsa may mahitabo sa imong host, unsa may mahitabo ani niya basta ako makasurvive ray. Nanay lahang mindset mapaparasite, mapabacteria ba na siya or mapavirus or fungi, no? Mga pathogenic ha. Ilahang goal is to survive. Okay, so your viruses are no exception. Ilahang goal is to survive and to proliferate, all right, and to produce more offsprings para mas mudaghan sila. Okay, all right, ayan. And that's for the steps of viral replication. Again, Apumar. Ayan, ato ang mnemonic. Okay, alright. So I hope na-gets lang to siya. Alright, now we go na to viral classification. So how do we classify the viruses? No, there are a lot of methods. But the most common good is through nucleic acid composition. Take note of that, dears talaga. How do we classify viruses? Number one is through nucleic acid composition. So, are they RNA ba? Are they DNA viruses? So, dira pa lang daan, mas-separate na nato ato ang mga viruses. It's usually through this description or through this classification na mas consistent siya. O, sana all. Because again, we know already that these are RNA viruses. Okay, these are the DNA viruses. So, dira pa lang daan, na-separate na nato ang mga viruses na maka-infect to humans. Alright? Now, again, single or double-stranded, as we have mentioned na ganina sa macromolecular synthesis, are they positive sense or negative sense ba? Alright. Next, we can also classify them according to their capsid. No? Are they helical ba? Kato, murag spherical iyahang shape, or icosahedral, murag siya cube-like iyahang shape. Next, the presence or absence of envelope. Again, if na-envelope ang virus, we term them as enveloped viruses. And for those na wala, they are known as the naked na viruses. Alright, okay. Now, are they acquired by from nuclear membrane, such as your herpes viridae, or those that come from the cytoplasmic membrane? So, ayan. And of course, as we have mentioned, the phospholipid bilayer, which is the envelope, they are susceptible to a lot of changes in the environment and detergents. And they're also ether sensitive. Alright? Ether sensitive. Of course, because polar, no? Polar molecules, they attract each other, no? Like charges attract. Okay. Also, they can be classified according to their disease, no? In terms of unsa may ang disease na cause, are they kanang respiratory ba na mga diseases, encephalitis no sa brain, enteric ba siya ng mga viruses, target tissue, again asa siya nag-infect sa brain ba, sa heart, no, and also means of transmission or vector, are they droplet, airborne, are they through sex ba, sexually transmitted, are they arbo virus, arthropod-borne na mga virus. Ayan. So, that's how we classify. And the most consistent is again through physical and biochemical characteristics such as their size, morphology, the type of genome, very important, and means of replication. I think nato'y recall sa boards, nag-i-ask how are viruses classified. So, take note of that. Pangitaag yun dayon ang nucleic acid classification because dira pa lang daan press the buzzer na agad-agad. Pero again, you look at the different choices pod. If na ay mga presence or absence of envelope, pwede pod, no? Morphology, pwede pod. Pero if wala gani, nucleic acid classification jud dears. Ha, please take note. Kana gyud bullseye answer gyud nato na nucleic acid classification. All right, okay. So that's for viral classification. Again, emphasis nako ha. The genome of the virus, again, genome of viruses only have either RNA or DNA and never both. Okay? Either RNA or DNA. Usara gyud. Dili pwede ang both. Or wala, dili ang duha na. Okay. Alright. Ayan, sige. Now, we go na to viral genetics. Okay, so these viral genetics, ano lang mga terms lang siya, dear. So, of course, as we have mentioned, diba, through the assembly of mga new virions, there could be errors na may tabo, no? Sa pag-replicate pa lang daan po mismo, there could be a lot of errors, no? And these errors could arise or could give rise to a lot of what we call mutations, alright? And very much common, especially sa COVID-19 karon, di ba? There are a lot of variants of your SARS-CoV-2, okay? And that is expected because that is normal in their life cycle, di ba? It's normal to have errors, no? Kita man gani masayop, unsa na nagyang mga viruses, di ba? So they are not foolproof, no? They are not error-free in terms of their replication. So there could be a lot of mutations yun. And these are again spontaneous usually, all right? That readily occur in your viral genomes, and these mutations again could either be for the good or for the bad, okay, for the better or for the worse, because again these mutations could be or could produce mga less infectious, less na mga severe na strains, or on the other hand they could also produce the more severe forms, more infectious forms, no, para sa again COVID-19, such as your Delta variant. Okay, now we go on to different types of mutations. You have the lethal mutations, so mutations that inactivate essential genes, meaning pag produce nila ni mutate, no, or na change gamay, na tweak iyahang pag-replicate, nawala noon itong mga essential genes na kailangan niya. So, mahitabo, mamatay noon itong mga bagong virions, no? Munang lethal mutations. Next, for deletion mutant, loss or selective removal lang of a portion of the genome and the function it encodes. So, delete jud tanan para sa imong lethal mutation. Gamay lang, portion lang, deletion mutant. Next, your plaque mutants, they differ from the wild type or the normal virus or parenteral, kung asa, katod yung original, kung asa nag-start, ayan. They differ from the wild type in terms of the size or appearance of the infected cells. So, sa host cell mo differ in terms of their size and appearance of the host cell nag-infect. Next, you have host range mutants. They differ in the tissue type or species of target cell that can be infected. Attenuated mutants, these are those that are, as I mentioned, they cause less serious disease in animals or humans. You have conditional mutants in terms of mga temperature, no? Those new strains that they become more temperature sensitive or cold sensitive. Ayan. These are new viruses that can only be produced at a certain temperature. Ayan. And lastly, recombination. This is an interaction between the genome of one or two different viruses, all right, or the interaction of the virus to the host cell na genome, all right. Example of that, if may mga co-infections between two viruses such as an HSV or a CMV, so pwedeng mag-merge ilahang genome, so mo-produce sila new strain, all right, para sa SINBs na virus and also the Eastern equine encephalitis na virus, nag-merge sila to produce the Western equine encephalitis inana. So that's another type of recombination. So nag-mix, no, ang duha ka genomes of different viruses, then they produce a new strain. All right, so that's for the viral genetics. Again, mutations are very much common, no, in viruses. It's really part of their life cycle. So dili nila sala na na mutation because it's part of their life cycle. It's expected, no, na na mutation, especially sa COVID-19. Munang daghang variants because again, different different hosts, no, different host cells, different host environments. So the virus will adapt to the different environment. So of course, na mga mutations na mahitabo, that's why daghang variants na nahitabo. So the more people will be infected, the more variants will happen. That's why si Dr. Anthony Fauci sa New York or sa US, of course, iyahang advice is of course to limit or to stop the spread of the virus, to prevent the production of more variants, because it's through the spread or it's through the interaction of more people or through the spread of the virus na mas daghang variants na ma-produce. Because again, different people, different host cells, different host environment, different host nutrients and all that. So the virus will adapt, no? So mas daghan pong changes na mahitabo sa virus, no? Mas daghan pong mutations ang pwedeng mahitabo. That's why there are variants na natong nakita karon sa imuhang COVID-19. Okay? Alright. Asa imong SARS-CoV-2 na virus. Alright. Okay. So that's for viral genetics. Okay. Alright. Ayan. Sige. Now, for the next video, we'll continue now with specimen collection for virology. So I'll see you on the continuation, on the next video. Alright.